Imagine for a second typing a sentence in plain English into your computer. Something like, design a cure for this specific mutation of lung cancer. And having the AI natively spit out the exact, 3D blueprint for a drug that has never existed.
Right, which sounds totally crazy.
Yeah, exactly. No translational software, no middlemen, just an artificial intelligence speaking the physical, three-dimensional language of biology as fluently as you and I are speaking right now.
It genuinely sounds like science fiction. I mean, it really does. But that exact capability is the horizon we are looking at today. The sources you brought us are detailing a fundamental shift in how machines understand the building blocks of life.
And that is our mission for this deep dive into the source material. We're looking at a groundbreaking new paper outlining a model called BioMatrix. The study itself is titled Towards a Comprehensive Biological Foundation Model Spanning the Modality Matrix of Sequences, Structures, and Language.
It's quite a mouthful.
It is. But, okay, let's unpack this. What researchers are attempting here is to teach AI to unify three totally different worlds. So you have 1D biological sequences, complex 3D physical structures, and then just standard human text.
Right.
For you listening, this is critical because, reading and writing biology natively is basically the holy grail for speeding up drug discovery and protein engineering. We are talking about shrinking decades of trial and error down to a single computational thought process.
To really appreciate how big of a leap this is, though, we have to look at the severe limitations of the models that came before it.
Yeah.
Up until now, biological foundation models suffered from a sort of, an architectural split personality.
Oh, that's a good way to put it.
Yeah, they were forced to choose between two highly compromised paths.
Right. I noticed that in the text. It said previous models either chased native multimodality or they chased broad entity coverage. But what does that actually mean in practice, like for the actual researchers using them?
Let's break it down. So native multimodality means the AI could handle a sequence, a 3D structure, and text all at once.
Okay.
But the catch was, it could only do this for one specific type of biological entity, just small molecules, for example. So the AI was deep, but incredibly narrow.
I see.
The other path, broad entity coverage, meant the AI could process molecules and proteins and DNA. That's a wide net.
Right, covering all the bases.
Exactly. But the trade-off was that these models completely lacked the ability to process human text or explicit 3D structures. So they were wide, but very shallow.
So to fix that, I was reading that a lot of those older, broader models use something called an adapter-based design to try and process everything. I've been trying to wrap my head around that.
It's a bit clunky, honestly.
Yeah. It feels like having an incredibly talented friend who can read complex sheet music perfectly in their head, but they don't actually own an instrument to play it back for you.
That is a brilliant way to look at it.
Like, they can read the incoming biological data, but they are trapped by their own output limitations because they have to use standard English text to answer you. They can't natively generate a 3D biological structure.
That sheet music analogy captures the exact bottleneck in AI biology over the last few years. The technical term for it is input-output asymmetry.
Input-output asymmetry.
You got it.
Yeah. These older models could ingest continuous, highly complex features from specialized biological encoders. But when it was time to generate an answer, they were forced to emit their outputs through a standard large language model text vocabulary.
Wait, hold on. Why is that such a problem, though? If the AI knows what the molecule looks like, why can't it just describe it to us using its text vocabulary?
Because you cannot easily describe the precise microscopic 3D spatial geometry of a complex folded protein using just the words in the English dictionary.
Right. I guess curvy and bummy don't really cut it.
Yeah.
Exactly. English wasn't designed to communicate atomic coordinates. It is fundamentally the wrong tool for the job. You lose a massive amount of physical information trying to cram a 3D biological structure into words.
So the BioMatrix team looked at this bottleneck and basically decided they needed to build a completely new instrument.
They completely threw out the external encoders and those projection adapters, just tossed them. Instead, BioMatrix utilizes a single decoder-only architecture.
Okay.
They built this foundation on top of the Qwen3 language model, specifically using the 1.7 billion and 4 billion parameter variants. Then, They continually pre-trained it on a staggering 304.4 billion tokens of data.
304.4 billion tokens. I mean, just to
Wow.
give some context to you listening, a billion parameters is actually quite small and efficient compared to the massive 70 or 100 billion parameter models out there. But training it on 300 billion tokens of biological data is wild.
It's a massive undertaking.
But the sheer scale isn't even the part that stuck out to me the most, is how they bypass that adapter bottleneck. They basically created a universal translator, right? Like a shared alphabet.
The shared discrete token space is really the core innovation of the entire paper. Since the fundamental problem was the inability to output non-text modalities, the BioMatrix team decided to put everything, the English text, the 1D sequences, and the 3D structures, into the exact same stream of tokens.
So it's all just one language to the computer.
Precisely. To the AI, a word like "the" and a 3D fold of a protein are processed using the exact same underlying mathematical logic.
I'm having a really hard time visualizing this, honestly.
It's tricky to conceptualize.
Yeah, like how do you actually turn a 3D squiggly protein or a complex molecule into a text-like token without completely losing its physical shape? To me, that sounds like trying to describe a beautiful three-dimensional marble sculpture using only a flat 2D Excel spreadsheet, you know?
Yeah.
You are going to lose all the curves.
It is a massive challenge, and it brings us to a piece of technology called a vector quantized variational autoencoder.
Wow, okay.
Yeah, or VQVAE for short. Let's look at how they apply this to small molecules first, using their molecule 3D tokenizer, which they call Molstructalk.
Molstructalk. Catchy.
Right. The brilliance of this tokenizer is that it completely ignores absolute Cartesian coordinates.
Cartesian coordinates, meaning it doesn't care where the molecule is sitting in an arbitrary room. Like, it's not mapping an XYZ grid of a static space.
Correct. The model doesn't care if the molecule is upside down or in the corner of a simulated box, because the molecule's chemical properties don't change based on its orientation.
Right, that makes total sense. A chair is a chair, even if it's upside down.
Exactly. Instead, Molstructalk captures relative local geometry. It looks at a single atom and measures its relationship to a neighboring focal atom.
Okay, so just relative to its immediate buddy.
Yes. Specifically, it measures the distance between them, the polar angle, and the azimuth. It essentially creates a localized spherical frame of reference for every single atom.
Wait, let me visualize this. Polar angle and azimuth. We are basically talking about longitude and latitude, but applied to the microscopic space between atoms, right?
That is a perfect way to think about it. It's recording how the atoms sit relative to their immediate neighbors. So it's saying, I am exactly this far from you at this specific latitude and longitude.
Oh, wow.
And once the model takes those geometric measurements, it maps them to a specialized codebook that contains 512 entries.
Just 512.
Just 512. It finds the closest match in that codebook and assigns it a discrete number. It fuses the chemical identity of the atom and its local geometry into a single token that the language model can now easily read and write.
I was looking at the methodology for that part, and the paper mentions they had to modify the decoder into something called a branch decoupled variant. I want to spend a second on this because it seems like a very deliberate engineering choice.
Oh, absolutely. It was crucial.
Yeah, they found that if they tried to predict all those measurements, the distance, the angle, the latitude, using one shared neural network pathway, the measurements actually interfered with each other. But why would they interfere? To a computer, aren't they all just numbers?
You'd think so.
Yeah.
But think about the actual physics of what the AI is trying to predict.
Okay.
Predicting a distance means guessing a positive length, which can theoretically stretch outward indefinitely. But predicting an angle is fundamentally different. It is bounded within a 360-degree circle.
Oh, right. It resets after 360.
Exactly. And predicting a binary sign, whether an angle is positive or negative, is a totally different mathematical task again. If you force a single neural network trunk to guess all of those wildly heterogeneous targets at the same time, the network just gets confused.
Yeah.
The data types clash.
Ah, so it's like asking someone to simultaneously solve a math equation, paint a picture, and sing a song.
That's a great analogy.
Like, they are all outputs, but they require totally different parts of the brain.
Which is exactly why decoupling the branches was so effective. Each of those structural targets gets its own dedicated prediction head. By stopping the different geometric measurements from fighting for the same computational resources, their structural accuracy improved significantly.
That clears it up immensely. Now, that process is for small molecules. But proteins are a completely different beast, right? They are massively larger and more complex.
Much, much larger. For proteins, BioMatrix uses a different tokenizer entirely called GCPVQVAE.
Another acronym to remember.
Right. And instead of assigning a token to every single tiny atom, it assigns one token per residue, which is a single amino acid. Because the structural variations in proteins are so vast, the 512-entry codebook isn't nearly enough. They had to use a 4096-entry codebook.
And the source note's a really interesting departure here. For the molecules we just talked about, they fuse the chemical identity and the geometry into one single token.
Right, the all-in-one token.
Yeah. But for proteins, they keep the amino acid token and the 3D structure token completely separate. The paper calls it a factored representation. If fusing them worked so well for molecules, why change the strategy for proteins?
It comes down to a mathematical problem called vocabulary explosion.
Sounds dramatic.
It is, for a computer. Let's do the math. If you try to create a unique, fused token for every possible combination of the 20 standard amino acids in the 4096 structural codes, you would need a dictionary of nearly 82,000 joint entries.
Okay, I see where this is going. If the dictionary is 82,000 words long, the AI is going to have a hard time learning the definitions of all of them.
Especially the rare combinations. The language model would barely ever see the tail end of those 82,000 combinations during its training phase. It simply wouldn't have enough exposure to learn them properly.
Right, it's like trying to learn English, but mostly just studying words from the 17th century.
Precisely. By factoring them, keeping the amino acid sequence and the structural fold as two separate but parallel token streams, the AI only has to master a dictionary of 20 items and a dictionary of 4096 items.
Oh, that's way more manageable.
Much more.
Yeah.
It sees all of those individual tokens constantly, allowing it to learn the sequence identity and the structural fold independently but simultaneously.
Okay, so if separating them works so well, how does the model actually know what to physically build in the real world? We have this universal alphabet now. Text, molecule tokens, protein tokens, all swimming in the same data stream.
It's all in the mix together.
Yeah. Which brings us to how the model reads the 1D sequences of molecules. And I found this part of the study genuinely surprising. BioMatrix learned two different dialects for 1D molecules, and they behave in completely opposite ways.
Yes, we're talking about smiles and selfies.
Two more great names.
Right. These are two heavily used line notations in chemistry. They are essentially ways to represent complex molecular structures as a single string of text characters. BioMatrix natively supports both dialects. But what the researchers discovered is that they are entirely complementary. They serve vastly different cognitive purposes for the AI.
I was looking at these benchmark results, and I was honestly stumped at first.
They are a bit counterintuitive.
Very. Because when it comes to open-ended optimization tasks like asking the AI to make a molecule more soluble or optimizing it for a specific drug likeness property called QED, the selfies dialect absolutely dominates the benchmarks.
It blows smiles out of the water there.
Totally. But then on tasks that require customized rigid constraints, like asking the AI, hey, build me a molecule with exactly 3 double bonds, selfies collapses to a near zero success rate, and smiles easily takes the crown.
It really is a fascinating split in capability.
If selfies is so incredibly smart that it dominates optimization, how does it completely fail at a basic task like counting 3 double bonds? It feels like a genius failing basic arithmetic.
It does seem contradictory until you look at how the design of the language dictates its utility. You see, selfies was specifically engineered by scientists to guarantee syntactic validity.
Syntactic validity, meaning what exactly?
The rule of selfies is that no matter how randomly you arrange its tokens, it will almost always decode into a chemically valid, physically possible molecule. It abstracts away all the complex surface form bookkeeping.
Meaning the AI can wildly hallucinate and experiment without breaking the laws of physics.
Precisely that. For optimization, where the model needs to freely explore the vast unknown chemical space and subtly shift properties, selfies is the perfect dialect. It provides a safe sandbox where the AI literally can't accidentally create an invalid, broken molecule.
But that exact same superpower is its fatal flaw for counting constraints. The paper gave this fantastic example of a benzene ring.
Ah, yes, the benzene ring example.
Yeah, if you write a benzene ring using the smile dialect, it is always spelled clearly, exactly the same way. Lowercase c1c c1. It is a distinct, recognizable word. But in selfies, the spelling literally changes depending on what is sitting next to it.
That is the concept of surface form instability. In selfies, if that benzene ring is part of benzoic acid, it appears as a contiguous block of tokens. But if you move that exact same ring into a molecule of biphenyl, the selfie's token sequence physically splits the ring apart and wraps it inside branches.
So it's constantly shape-shifting?
Exactly. Furthermore, selfies folds bond types directly into neighboring atom tokens. A triple bond isn't a standalone character you can just point to. Its existence is baked into the token of the atom right next to it.
Let me see if I have this straight. If I tell the selfies model, give me exactly 2 double bonds, it looks at its own internal vocabulary and says, I don't actually have a dedicated token for a double bond, and my benzene rings keep changing their spelling based on the context. I have no anchor to count this.
That is exactly the failure mechanism. The AI cannot anchor specific structural constraints to recognizable token patterns because those patterns are entirely context-dependent in selfies.
What?
Smiles, on the other hand, is explicit and rigid. A double bond is always represented by an equals sign. A triple bond is always represented by a hashtag. It is incredibly easy for the language model to just count those explicit characters.
It's like using beautiful, flowing poetry to convey a complex emotion, but needing a rigid legal contract to buy a house. You can't buy a house with a poem, and a contract makes for terrible art. You need both types of language.
That's a really lovely way to put it.
The fact that BioMatrix kept both languages as a strategic feature, allowing the model to switch dialects depending on the specific cognitive task, is just brilliant.
It highlights a deep understanding of how language shapes thought, even for an artificial intelligence.
Okay, so the 1D text space is working beautifully, but the ultimate test of BioMatrix, the whole reason they went through the trouble of tokenizing 3D space, is whether this language proficiency translates to generating accurate 3D structures in the real world.
Right, the physical application.
Exactly. How does it handle cross-modal interactions?
Well, the benchmark results here are staggering, especially given the model's size. BioMatrix achieves state-of-the-art or highly competitive performance on 77 out of 80 biological tasks.
77 out of 80.
It's remarkable.
That covers single-entity tasks, multi-entity tasks, deep biological understanding, and physical generation, all across and within different modalities. Again, for you listening, we are talking about a highly efficient 1.7 billion and 4 billion parameter model going toe-to-toe with and beating models that are massively larger and hyper-specialized for single tasks.
The cross-modal triumphs are really where the unified architecture flexes its muscles. Take property condition 3D generation. This is a task where you give the model a target quantum chemical property, like a specific HOMO energy level, and ask it to build a 3D molecule from scratch that exhibits that exact property.
Just to clarify for anyone who isn't a quantum chemist, HOMO stands for highest occupied molecular orbital. It's a crucial measurement for understanding how a molecule will react chemically, essentially mapping its electron behavior.
Right. And on these incredibly complex electronic structure targets, BioMatrix reduced the mean absolute error by roughly 3 to 4 times over the strongest prior method.
3 to 4 times.
Yes. The previous state-of-the-art was a highly specialized diffusion model called NextMol. Specifically, BioMatrix dropped the error for HOMO energy from 205 millielectron volts down to just 53.
Dropping an error rate by a factor of 4 isn't just an incremental improvement. That is a completely different tier of precision. But in the spirit of full transparency, the paper did point out a flaw when it came to 3D generation.
Yes, it did. It's not perfectly flawless.
On unconditional 3D generation, which is basically asking the AI to hallucinate a physically viable 3D molecule completely from scratch without a specific target, it perfectly matched the dihedral angles, but it had a higher error rate in fine-grained bond lengths compared to those older diffusion models.
That's right.
So if it's making mistakes on the actual microscopic length of the bonds connecting the atoms, is the entire model broken? I mean, how big of a deal is that?
It's not broken at all, actually. What you are looking at is a known inherent limitation of the tokenization approach itself. The researchers refer to it as irreducible quantization error.
Irreducible quantization error.
Yes. Remember that 512 entry code book we talked about earlier?
Mm-hmm.
The universal translator for the local geometry?
Yes, the dictionary that maps all the distances and angles.
Think of it like painting a highly detailed photorealistic picture, but you are only given a palette with 512 specific colors. You can get incredibly close to the true image, but some of the most subtle shades, the exact microscopic fraction of an angstrom for certain bond lengths, are going to get slightly routed off to the nearest available color in your palette.
Ah, I see. So it's not that the AI fundamentally misunderstands the chemistry of the bond. It's that its vocabulary simply isn't granular enough to express that exact infinitesimal fraction of distance.
Exactly. Furthermore, BioMatrix generates these atoms one by one, autoregressively. It places atom A, then atom B, then atom C.
Right, like typing a sentence word by word.
Exactly like that. If it makes a microscopic rounding error on atom A due to that limited palette, that error can slightly compound and push atom D slightly further out of alignment down the line. Diffusion models, by contrast, don't work step by step. They denoise all the coordinates globally at the exact same time, so they don't suffer from that sequential compounding error.
But the paper notes there is a really elegant, easy fix for this compounding error.
Yes, there is. The researchers apply a single, highly lightweight post-hoc correction called an MMFF refinement.
MFF, that's a standard physics-based force field, right?
Yes, you can think of it as a set of classic physics rules that know exactly how far apart atoms actually want to be based on their natural energy states.
Okay, so it just runs a quick check.
Pretty much. They take the model's slightly rounded off 3D structure and run it through this force field. It lets the molecule naturally relax into its mathematically perfect lowest energy bond lengths. It takes a fraction of a second. And once they applied that quick refinement, the gap in geometric accuracy between BioMatrix and the diffusion models almost completely vanished.
So it's like giving the molecule a quick digital massage, letting all the joints snap into their natural mathematical shape.
That's a fun way to picture it, yes.
You accept a tiny bit of rounding error at the token level, which you can fix instantly in exchange for the massive power of having a single, unified AI that can translate English text directly into 3D molecular biology.
Exactly.
That trade-off seems absolutely worth it.
It is a phenomenal trade-off. It definitively proves that next token modeling, the exact same autoregressive mechanism that predicts the next word in your text messages or writes an email, can generate highly valid, physically plausible 3D biological structures, provided you give it the right tokenized vocabulary.
Well, let's pull all of this together because the journey of this paper is incredible. We started with the bottleneck. Older AI models were trapped. They were forced to use external adapters that couldn't natively write the biology they were reading.
Right.
BioMatrix shattered that bottleneck by tokenizing 3D space itself. They turned local geometry, the latitudes and longitudes of atoms, into discrete tokens, allowing a single language model to read and write 1D sequences, 3D structures, and English text simultaneously.
And we saw how it smartly leverages the unique strengths of different chemical dialects, too. Using selfies for open-ended optimization because of its safe syntactic validity, and switching to smiles for rigid, customized counting constraints because of its surface form stability.
And the benchmark victories really speak for themselves. I mean, BioMatrix proves that a natively multimodal generalist model, even running at a highly efficient 1.7 or 4 billion parameters, can match or outright beat the hyper-specialized 8 billion parameter models. It is predicting and generating the molecular and protein structures that will literally drive the medicines of tomorrow.
Which opens up a fascinating philosophical and technological horizon for us to think about. We'll consider this. We've just seen that the complex, three-dimensional physical geometries of proteins and molecules can be mathematically flattened into the exact same type of discrete tokens that make up human speech.
Right.
If the fundamental architecture of language models can learn to seamlessly translate between English prose and microscopic spatial reality, what happens when we start adding completely new, massive modalities into this exact same token stream?
Oh, wow. Like what?
Could we eventually tokenize the biomechanics of entire human cells, or the interactions of human organ systems?
That is mind-boggling.
Just imagine an AI that can effectively read a patient's medical chart in English and natively write a custom biologically simulated cure, all in a single fluid computational thought process.
That is exactly the kind of question that makes this field so incredible to watch. We are moving from predicting text on a screen to predicting the physical building blocks of life itself. For everyone listening, I hope this deep dive gave you a new perspective on where AI is heading. Keep wondering, keep learning, and keep looking for those unseen connections that change how we understand the world. Until next time.